Hello everyone. This section we will study the point about pathophysiology of cancer, part 2. Tumor suppressor genes may function as the brakes of inhibiting cell growth, fixing broken DNA, or causing a cell to die. These types of tumor suppressor genes can be thought of as gatekeeper genes. Yet, some tumor suppressor genes function in more of a caretaker role. These genes create proteins that oversee and regulate many of the functions of other genes to maintain the stability of DNA. RB, APC, and protein 53 function as gatekeepers. In contrast, BRCA1, BRCA2 genes function more as the caretakers and regulate the activity of other proteins involved in cell growth and repair. In our body, we have two sets of chromosomes and two sets of genes, one from each of our parents. With dominant genes, only one of the two copies need to be mutated or abnormal for a negative effect to occur. In general, oncogenes are dominant. Tumor suppressor genes, in contrast, tend to be recessive. That is, two suppressor genes must both be damaged in order to contribute to cancer. Examples are the BRCA1, BRCA2 genes. People who have a mutation in one of these genes have an increased risk of developing breast cancer. However, not everyone with the gene develops breast cancer. The first copy of these genes is mutated at birth, but it's not until another mutation occurs after birth with an acquired mutation or somatic mutation that abnormal repair proteins are made that increase the risk of cancer. This recessive nature is what is referred to in the two-hit hypothesis of cancer. The first copy mutation is the first hit, and a later mutation in the other copy is the second hit. Another characteristic of tumor suppressor gene is haploinsufficiency. Haploinsufficiency of tumor suppressor genes, wherein the reduced production and activity of proteins result in the inability of the cell to maintain normal cellular function, is one among the various causes of cancer. Representative cases are protein 27 KEAP1, protein 53, DMP1, NF1, and PTEN. Some tumor suppressor genes are in a sequence in which a mutation occurs that results in a gene product adversely affecting wild-type gene products that are all in the same cell. It is called the dominant negative effect. Example is the mutation in protein 53 that results in cancer alter the function of the wild type. Normal protein 53 works for cell cycle arrest, DNA repair, and, if necessary, apoptosis and DNA damage, cell cycle abnormalities, and hypoxia. Mutated protein 53 can cause continuous cell cycle and cancerous cells. Tumor suppressor gene inactivation indicates a complex view. The possible inactivation ways include gene mutation, loss of heterozygosity, and the hypermethylation of gene promoter region, which belongs to epigenetics introduced in topic 2. A cancer that has spread from the place where it first started to another place in the body is called metastatic cancer. The process by which cancer cells spread to other parts of the body is called metastasis. In metastasis, cancer cells break away from where they first formed, that is primary cancer, travel through the blood or lymph system and form new tumors in other parts of the body. That is metastatic tumors. The metastatic tumor is the same type of cancers as the primary tumor. Cancer cells may be able to influence the normal cells, nucleus, and blood vessels that surround and feed a tumor. This is the area known as the microenvironment. For instance, cancer cells can induce nearby normal cells to form blood vessels that supply tumors with oxygen and nutrients which they need to grow. These blood vessels also remove waste products from tumors. Cancer cells are also often able to evade the immune system. Although the immune system normally removes damaged or abnormal cells from the body, some cancer cells are able to hide from the immune system. Tumors can also use the immune system to stay alive and grow with the help of certain immune system cells that normally prevent a runaway immune response. In conclusion, the hallmarks of cancer include tumor-promoting inflammation, evading growth suppressors, avoiding immune destruction, sustaining proliferative signals, enabling replicative immortality, genomic instability and mutations, resisting cell death, activating invasion and metastasis, inducing angiogenesis, and degradation of cellular energetics. This is the end of this section. Thank you very much.